Yes, good evening. This is Dan Davidson. I'm the moderator for tonight's session, the fifth webinar in a series of 6 on soil health. This webinar is number 5. Its speaker is Will Brinton. I would introduce him in a moment. He will be speaking on CO2 respiration, nutrient cycling tonight. The webinar we had last week, scheduled for last week with Odette Menard, that's being rescheduled for next Monday night. You probably should have got an email today on that. There were some IT issues last Tuesday night that prevented us from getting on to the go to webinar. So we're sorry for that. For tonight's webinar, this is going to last about an hour or so. Feel free to post any questions in your question box, and I will forward those over to Dr. Brinton to answer. The sponsor for tonight's session is Ward Laboratories in Kearney, Nebraska. Ward Labs actually runs the Haney test, which includes the Solvita test. So we want to thank them for their sponsorship of tonight's event. Our speaker for tonight is Will Brinton from Maine, Woods End, Maine. Dr. Brinton runs his own lab, Wood End Labs, and he, along with, in conjunction with Rick Haney, developed the Solvita test that he'll talk about, and then working with Rick Haney, they developed a Haney test. And both of these are very important tools in measuring and monitoring soil health. So without further ado tonight, again, we thank you for coming online, and we ask you to go ahead and post your questions on through the question box, and we'll turn this over to Will now. So Will, take it away.
Good evening, and thank you, Dan, for your efforts to pull this together. It's a hot evening here in Maine, and I expect it is elsewhere in the country. I'm very pleased to be able to have the opportunity to give this presentation, and as Dan indicated, please post your questions and we'll try to answer them at the end of the session. We are a laboratory specializing in soil mineralization and biodegradation studies. And on this particular topic, my view is that soil respiration as such implicates the entire soil plant system and should not be seen in isolation. Although in convenience, we will analyze individual traits and try to bring them into context with the whole topic. The exciting thing about this field is that it continues to grow. And from a big picture perspective, we have this comment I want to begin with. Respiration is a critical determinant of the carbon balance at scales as familiar as the vegetative stand, as politically relevant as the European continent, and as ultimately important as the globe. Proceeding from this to our theme tonight, coming more down to earth, I want to concern myself and present to you my views on soil CO2 respiration in an agricultural context. There are many cycles here interconnected, each one of which is a field in itself. We basically are starting here with the concept of the supply and turnover of soil organic matter. This is mediated by micro and macroorganisms throughout the process. If the soil is not frozen, anywhere from 3 to 4 degrees Celsius up to 50 to 60 Celsius is the range of activity of most microorganisms in the soil. And during this whole cycle, we have the episodic release of carbon dioxide from the microbial consumption of soil organic matter. And simultaneously, we have this cycle furnishing plants with mineral nutrients released from the carbon bonds that they have been held in, and at the same time provisioning CO2 for plant assimilation. This, therefore, gives an indication of how large this cycle of respiration is. From my perspective, the preeminence of CO2 as a active, multi-diverse agent, and the microbial redundancy underlying its production in soil systems suggests that CO2 and its cycling exists to serve higher plant kingdoms, as I will show. We will go into some details on different aspects of this during the presentation. One thing I want to emphasize is this is not a new field. My training under Albert Schatz took me to Europe, and I studied under some of the personalities that I've asterisked in this chart here. This involved me in a historical study of soil respiration, and it's very noteworthy that at the same time that the Liebig era of nutrient mineral theory, Liebig, Mitscherlich, Gilbert, and Laws was developing in the late 19th century, a complementary movement with names such as Volney, Severin in Germany, Stoklasa in Czechoslovakia were developing a view and theory on the atmosphere of CO2 in soils. These events were obviously interrupted by two wars. There was a steady progression, and in the era of the 20s, standing out as preeminent in developing the field of CO2 respiration as microbial fertility was Lundegårdh in Sweden, who worked at Uppsala near where I later studied. And his work influenced a lot of other people who picked up the trail, including Dr. Voxman at Rutgers in New Jersey, who in the 30s reproduced a significant amount of Lundegårdh's work. I have not found any trail after World War II of that effort at Rutgers by Voxman, but Albert Schatz was a partner there and showed me some of the early notes and the excitement of this emerging biological fertility field. In the U.S., we had Gainey in Kansas and Potter in Iowa, who wrote astonishingly complex and complete essays and research documents on the parallel formation of nitrogen as CO2 is respired from soils and the implication for soil fertility. This work was also picked up in the UK by Russell, a well-known name in the line of textbooks on soil conditions and plant growth, and continued on into the present. Jumping post-war, post-World War II, was a whole new era of development, centered partly in Germany, Kenya, France, Switzerland. All these workers in the 50s into the 70s took up the issue of biological measurements of farming system effects. The work I did under Kerpf, who did his doctorate in Germany on CO2 respiration, and Pedersen, who was a Swede who did long-term field biology studies, influenced my choices of methods later. But as we can see, and what I am saying is that we had a fully developed institution of soil biology studies by the 1980s. It was well developed, and the indicators and methods were all essentially worked out. The current era now is very exciting to me because this work by many early people underscoring and supporting biological farming movements has now been expanded to include all farming. And I'm particularly excited to see the new interest coming from all sectors of agriculture and biological indicators of fertility. Well, this long history of development left with it a trail of methods for doing CO2 respiration studies. These are all examples of this installed in my laboratory, and we are familiar with them all. I'll just briefly list them all. The interesting thing about these methods, despite some of them being over 100 years old, they're all still being used today. Base trap and visual potentiometric titrations, a method we used for quite a period of time before moving beyond that. Infrared methods exist. We use these for short-term studies, 1 to 3 day studies. They're limited by the small number of units you can assign to them. For long-term CO2 mineralization studies, we have a CO2 train apparatus like this. You can incubate 18 samples at once as large as 2 liters in volume. So you can do a longer-term study and capture all the CO2. Again, it's not intended for routine commercial testing. Gas chromatography is well recognized, and here we can separate oxygen from methane and CO2 during respiration studies and quantify them individually. Some of the oldest methods are manometric methods. This is a Harvard respirometer that was built in the 50s that we have and use in the laboratory here. Manometric methods were upgraded in Germany recently with the invention of the oxytop system, so that technology is still widely available for soil respiration studies. The ultimate approach is to do CO2 flux chamber, and we use these in field studies. This kind of apparatus is definitely research-only grade, and it takes a day or two to set it up in a field, and you can monitor one location but produce very, very interesting and detailed evidence of the efflux of CO2 under ambient conditions. Finally, obviously, one of the more recent developments in an entirely new approach to the chemistry was my development of the Solvita method, which I will describe in somewhat more detail. I want to point out the significance of CO2 respiration from 2 different perspectives. One is the perspective of its functional role. And I tend to believe today we take too narrowly focused a view or a methodology on respiration studies. One of the big pictures here is that CO2 provisions plants with CO2 for photosynthate. In other words, the respiration from the soil is directly implicated in photosynthate demand. The other interesting thing that tends to be overlooked is that respiration maintains the soil CO2 atmosphere. And if you do studies, you find that the carbonic acid in the soil is anywhere from 0.02 to 0.1 molar at any given time. And the more lively a soil is, the higher the carbonic and carbonate system atmosphere present in the soil. Implicated, of course, is holding and releasing nutrients. So when we talk about respiration, we could be talking about immobilizing nitrogen as well as mineralizing nitrogen. You have to understand the carbon and nitrogen dynamic here to know which one it will be. And somewhat outside and above all this is the obvious fact that CO2 respiration represents the Earth's major carbon sink source cycling phenomena. Now, from a parameter point of view, from a laboratory analysis point of view, we are all using CO2 as an indicator of overall soil biology. And I would point out that you have in situ and ex situ. Some ecologists refer to above ground respiration and below ground respiration. But in situ, if you're doing field studies as we do, you have root respiration included with bacteria, fungi, and macrofauna respiration. Ex situ, in the laboratory, when we bring samples in, we're primarily measuring bacterial and fungal respiration. Now, one footnote here is clearly a background phenomena. Geological release of carbonate CO2 does exist, and chemical weathering of minerals actually consume CO2. So these are competing inorganic CO2 reactions that exist at a trace level in soils. And the only interference we observe from them is if we try to measure respiration immediately after liming, where you will pick up CO2 coming off the carbonate decomposition. I want to go back to Lundegårdh's model because he's the one that really clued me on earlier to the significant, great role that CO2 respiration plays in the soil plant system. So he started by quantifying that plants have specific CO2 assimilatory requirements defined by the photosynthetic production system. Soils also have a specific CO2 respiratory yield. So this is taking place right around and beneath the plants. On top of this, plant roots have their own respiration. It's classed sometimes as nighttime respiration, but It's consumption of oxygen and production of CO2 that fills up the soil voids with additional CO2 products. Finally, Lundegårdh put all this together and said, you've got to look at the whole system. The plant assimilatory CO2 demand minus what's being produced by the soils and the roots indicates the carbon quotient for a soil-plant system. And obviously what he did was go further than this and define under full crop growth conditions how much CO2 are these plants requiring per day. So wheat, which was quantified earlier on, can require up to 124 kilograms of CO2 per day over a 60-day assimilatory period. I calculated corn recently, and that quantity of consumption could be as high as 461 kilograms of CO2 per day per hectare. So this is quite a significant amount of CO2 that the plants are requiring. So again, without soil CO2 respiration, where does all that come from? It comes from the air. And you can quantify the cubic volume of air that plants need to filter to provide all the CO2. These calculations show that wheat would require approximately 10 cubic acres or 10 cubic hectares, respectively, to supply 1 surface acre of crop growth over the 60 most intensive days of growth. In corn, 38 cubic acres of air would be implicated just to supply the carbon dioxide to the growing plant. This clearly is not necessarily the case, and Lundegårdh constructed rich soil, poor soil scenarios which fascinated Voxman at Rutgers, and I've seen him in some of his early writings. He redrew some of these diagrams. From Lundegårdh's perception, a low fertile soil has a low CO2 yield. In this example, 30 kilograms per hectare per day. I will show you later in the way we do basal CO2 respiration, that would be a test level of 3 parts per million in terms of the quantitation at the soil level. Now, on this scenario, a plant would have to get most of its CO2 from the air because the soil is not able to furnish any more than a small fraction of it. In a humus-rich soil, which they carefully measured and documented, you can have CO2 yields easily of 125 kilograms per day. This, in a basal respiration test, this would be a yield of 11 parts per million. And under this scenario, a full-growing canopy of wheat would be completely supplied in its CO2 needs from the soil alone. So this is one of the astonishing facts of CO2 respiration that I think we have to just put there in the background as we try to understand in the bigger picture what we mean by soil health. There's other new current information that's very, very interesting that shows that nutrient uptake, in particular nitrogen utilization, comes at a great CO2 cost. Now, traditionally we talk about growth respiration and maintenance respiration in plants, but new plant physiological studies show there's a whole ion uptake respiration scenario involved here. So that from the perspective of nutrient uptake, we often draw diagrams like this that suggest, although they do not say, that nutrients are just passively absorbed by plants. There's an actual negotiation. And research is showing that the uptake of anions, and particularly nitrate, is very costly to respiration. It involves as much as 50% of the maintenance respiration and possibly as much as 20% of the growth respiration, which directly implicates photosynthesis uptake of CO2 from soil and air. So this nutrient exchange is negotiated based on the sufficiency of cycling carbon, which means if we can look at this in the bigger picture, the plant is negotiating with soil to take up the nutrients based on the provisioning of carbon dioxide to the entire system. So briefly, my perspective on this, as a very young intern in Europe, I worked at the Swedish Soil Fertility Institute and was the one assigned to do respiration studies and these long-term plot studies. My advisor, Kerp, from Hohenheim University, actually did his doctorate in 1953 to standardize CO2 respiration. So he was naturally interested to get me deeply involved in that. Back in this country, my laboratory incorporated a 7-day respiration method that I was taught in Germany and included it in a nutrient audit with a nitrogen mineralization estimate associated with it. By 1995, we had developed the Solvita gel technology because of the limitations in the laboratory of these time-consuming methods or expensive technology that couldn't handle more than maybe 5 to 20 samples a day. It was after 2005, between that and 2010, that we had the innovation of the CO2 burst test, an adaptation of CO2 respiration, thanks to the work of Haney and Franz Lüber's research in the prior decade. This led to lab standardization of CO2 respiration, and now this method is included in the ALP and more recently in the NAPT for proficiency monitoring. In 2012, we introduced another detection method called soil labile amino N, or SLAN. It's a way of using a detector probe that's similar to these CO2 probes to pull out the amino N associated with humic extracts of soil. I will not be talking about that tonight. As Dan suggested at the beginning, from now, at this time on, we are working to incorporate these biological parameters into commercial soil testing methods. I want to give you a little bit of perspective on the CO2 probe system we developed. The basis of it was a simple detector system that obeys Beer's law. In other words, as the concentration of CO2 increases, there's a logarithmic response. We've created a linear scale of color, which is associated with a visual interpretation. The basis of this test was to create a system that was accurate with or without a spectrophotometer to read the results. And to this day, in our calibration and standardization, we maintain the consistency of the visual color chart with the calibration with the colorimeter that's much more precise, so that the test is as useful in the field, though less accurately so in terms of color reading than in the laboratory. One of the things we had to do to make a breakthrough in this arena to enable laboratories to be able to do it on a larger scale was develop a low-cost spectrophotometer, and we built this thing from the ground up based on the chemistry that we had captured. And we used a complex set, it's called a matrix array of photodiodes that can simultaneously filter 3 wavelengths of light with a tremendously broad dynamic range based on the optical outputs. So we can detect with this a range of 10 to 30,000 parts per million as carbon dioxide. I wanted to show you an example here of the way the system changes in a 24-hour period of time. These are 2 soils, and we're showing the color change up above, and at the end of 24 hours, this is a typical result. On the left, you have a soil with a basal respiration close to 20 parts per million, and on the right one around 6 parts per million. The visual chart can be recorded by photography or by reading in a digital colorimeter. As I've implied in this presentation, there are different applications for this. The more recent development leading to standardization in laboratories we call the CO2 burst. Some people also call it the drying-rewetting method. Drying-rewetting is a phenomena that was simultaneously reported in Africa and Europe in the mid-50s. In Africa, it was by Birch, and in Europe, it was by Kerpf, who I worked under, and a Swiss researcher. They had observed that soils subjected to drying when they are remoistened produce more CO2 than could be accounted for by basal respiration. Prior to this, we had developed a basal test, which is using as-is soil from the field under more normal conditions, that is, without drying and rewetting it. Both these tests are as valid as the other. The laboratory procedure enables much greater control and standardization. The soil is weighed, and this one it's filled to volume, so you have a less precise but a more typical result of actual field conditions. We are presently one of several labs that use this method within a overall soil biology type testing protocol, and some laboratories as a soil health test protocol. Basically, we're incubating samples. In our lab, we run all the tests at 24 Celsius. Samples come in from the drying and grinding oven in bags and go right into the jars. And the nice thing about our method is it has such a visual impact. You can lay them out and photograph a whole series of soil samples from a farm and quantitate them to the part per million level in the test process. I want to say a little bit about drying and rewetting because it's such a significant phenomena. This is, as I indicated, it was first observed in the 50s in Europe and Africa. If you go back and read the early work, it was particularly noteworthy that at the Kenya Ag Experiment Station, they had observed that growers and farmers depended on rainfall events that are sporadic during the season, and they seem to get enormous bursts of nutrients following these rainfall events. And they started characterizing what happens, and it came to be called either the birch effect, the priming effect, or more recently the soil CO2 burst. Now, this data here was recently published. It's work done in Germany with what's going on with priming in soils, and these are 4 scenarios that they have studied. The dotted line here represents basal respiration of soil as it would be if the soil was maintained at a given moisture content somewhere slightly under field capacity through this period of time. However, if the soil dries down, as you see here, this is a severe and deep drying event, and then rainfall returns or moisture returns, you have a burst of activity of CO2 here that reaches a magnitude that exceeds what you will ever observe with basal respiration. Now, they have found circumstances where if the drying is not significant enough, you get no CO2 burst. And last year, working with Dr. Franz Lubers, we quantified this as a necessary fact here is that soil has to be drier than 4% moisture to observe a CO2 burst. If it is higher than that moisture level, you will actually experience a basal respiration phenomenon, not a CO2 burst. Now, these following 2 scenarios have been studied just in terms of how low was the soil when it dried out and how dry did it get. You can experience some very significant bursts. The method we use in the laboratory, called the CO2 burst, is samples come in moist, they're dried over a 2-day period at approximately 40 Celsius and immediately re-moistened, and we experience this. There's quite a bit of discussion now, as evidenced in this chart down here. Does CO2 burst deliver more CO2 respiration, hence more nutrient release, than if the soil were kept moist steadily under basal respiration conditions? And the evidence, the jury is really still out on this. Under some scenarios, if these events are long and deep enough, basal respiration will still yield more overall mineralization than CO2 burst, but in some circumstances, it's the equivalent. It all depends on the time frame that takes place in between them. What is so convenient about CO2 bursts is we get enough CO2 in a short period of time to readily quantify it, and clearly it indicates the soil's priming potential. Now, what is nutrient mineralization? And I want to say a little bit about this. There's some very basic stoichiometric relationships that dictate respiration leading to a nutrient yield. And I indicated here in this first equation, basically, if you take the summation of CO2 as carbon coming off of organic matter and divide by the respective ratios of the carbon to the N to the P and to the S, and minus from this the system demand for the nutrient, you will have your respiration-based nutrient yield. Now, this ND, the nutrient demand, could be your crop residue decomposition factor, so that you might not yield any available nitrogen if at the same time significant carbon has entered the system, requiring nitrogen for its decay. Now, the method in Germany that I was brought up on was this formula, which is still published in the Schöchling and Bloom soil analysis textbook. They calculated nitrogen mineralization as the parts per million CO2 carbon divided by the percent of total carbon multiplied by the percent of total nitrogen times a factor for time. And this was an arbitrary factor where we would decide if a soil sample that was set up in respiration for 1 day, 3 days, or 7 days, what portion of the average soil respiration for the year would that represent? And we multiplied it out to obtain our nitrogen mineralization. Here are some examples of work I've done in collaboration with Dr. Rick Haney and Dr. Luke Baker at Brookside in quantifying these relationships using the CO2 burst method. In one of the studies we did with 257 soils, we compared the 1-day CO2 to the anaerobic nitrogen mineralization with the 7-day method and plotted it here. And you can see there's, you could say, almost a one-to-one relationship, asymptotic curve that looks like a yield dependency curve. As the respiration gets very high in soil, the rate of release of nitrogen through this alternative methods appears to decline somewhat. But we've experienced quite some good correlations with some of these other earlier methods. Now, when we've taken this to the field and compared the yield of control plots that had no additional nitrogen with the respiration rate, we get fairly satisfying relationships, but the intersect has to be determined. Here, 50 units of respiration correlated with 80 bushel yield, which we equated to 80 pounds of nitrogen. It could have been anywhere from 70 to 90. With wheat fields, this is data from Dr. Haney, who assembled control plot data from farms across the Oklahoma, Texas area and found this relationship, slightly over a one-to-one relationship, a soil CO2 respiration and the amount of nitrogen that was uptake by the crops. One thing that has to be stressed here in all this is the strong dependency on soil CO2 respiration of the temperature of the system. And there are two ways to account for this. The Arrhenius Swedish equation that dates to, I think, about 1880 is still the basic component here. Soil respiration responds to temperature in an exponential function. This is the original Arrhenius equation. You can plug in your figures and develop a curve of predicted CO2 release. And here is experimental data that was recently published showing a remarkably good fit if you look at the carbon dioxide efflux, and this is from soil flux chambers in relationship to the change of temperature from 10 to 20 to 30 degrees. We repeated this work recently in an incubation study using our Solvita test, and we were able to fit it fairly satisfactorily to an exponential curve that closely matches the prediction. What I am trying to say here is that whatever way you are measuring respiration is likely to be highly dependent on the temperature of the system. There's no general agreement what to use in the laboratory, but most of the studies are between 20 and 25 degrees. Anything higher than that would represent a much more unusual condition of soils, even though they do episodically attain very high temperatures in the summertime. So in extrapolating data to the field, we have to be aware when we do incubation studies in the laboratory, we have simulated a constant temperature environment, which is not the case under soil normal conditions. The other second factor that is large but not as large as temperature is the moisture role in CO2 respiration. Now, if you read the literature, you will find significant evidence for a very broad range of CO2 relative to moisture content of the soils. It tends to peak and then decline very slowly as soils reach higher and higher and exceed field capacity. We have been able to capture this in a simple way to produce the drying-rewetting phenomenon. This work was done by Haney and published in 2010. It uses a perforated beaker. There are holes in the bottom and a small glass fiber filter, and moisture is placed here, and capillary attraction drives it up, pulls it up into the soil, simulating field capacity moisture, and this is acquired in the space of less than a minute. So we're able to bring these soil samples rapidly to these moist conditions. If you stand back and look at moisture and temperature, you can understand the complexity of trying to predict a seasonal CO2 efflux going from laboratory to field. You have diurnal temperature swings that can be quite large. In fact, just looking at different layers of soil, the topsoil layer will experience more swings than soil between 5 centimeters and 15 centimeters deep, and so on. So you have different CO2 rates at all the different horizons. Now this data, which I took from the USD's climate analysis stations, shows two scenarios of Pennsylvania and the Palouse, Washington, with temperature peaking midsummer, both of them nearly the same. And interspersed in this are the rainfall events. As you can see here, it's wet in the spring and then dries down very severely. Then you have these bursts of moisture that could come close to being drying-rewetting phenomena, particularly in the Palouse where the soils dry down close to that 5% and lower mark. You have a real basis for predicting a significant nutrient flux from the birch effect or the priming effect as moisture returns. And moisture events probably drive the nutrient cycle under these kinds of circumstances. If we go out into the field and compare, this is a single farm study from samples taken recently by NRCS in Aroostook County. And these are fields across the farm and 2 depths of sampling. Now the way you read these colors is the brighter the color, the higher the respiration. So you can see quite a bit of variation going across this large field here. And each soil was paired with a subsoil equivalent. And you can see in virtually all cases the subsoil was had less respiration. But in one case here, we had a sample of subsoil that had greater respiration. This could have had to do with variations in the AB horizon, or it could have had to do with incorporation of organic matter. But again, this test is interesting in that you can quickly lay out your samples and have a strong visual impact. And we're working on a new technology where you could simply photograph all your probes and reproduce the color value and hence the respiration rate at any point later in the future with a reading device. I want to jump back into the past again and show what some of the early long-term studies that I did my master's work on revealed to us in terms of soil respiration effects relative to management practices. This was a study that I was involved with at the end of 17 years of treatments with 8 different plot systems, and they were comparing crop rotations and manuring practices. As you can see here, we had different versions of either raw manure combined with inorganic nutrients, composted, and a commercial organic form of compost, and 3 inorganic nutrient rates. And we measured all the enzyme levels, porosity, soil structure, earthworm canals, and 7-day and 1-day respiration in these plots. And you can see that over 17 years, these treatments really enhanced or certainly affected soil respiration. One of the things we found clearly that soil respiration indicated many biological effects. It correlated with just about any other biological factor from earthworms to dehydrogenase activity that was measured in the soil. And the other interesting thing was the higher crude protein yield that we characteristically obtained from the inorganic fertilizer plots was not reflected in increased soil biology. In other words, you see a disconnect here between nutrient supply and yield capacity and soil building that has to be taken into account. I recently recorrelated these data that I had from my early studies and found a stunning correlation between respiration rate measured either in the 24-hour scenario here or the 7-day scenario here. This is the way we did earthworm canals, or earthworm counts, as we counted all the canals after cutting down 10 centimeters and clearing off a square meter of soil. But this range here of about 20 to 150 is, for all purposes, the typical respiration range you will see when you do CO2 burst studies. With basal respiration, the levels are much lower. This is a 1-week respiration that included the initial burst of rewetting the soil, and then it levels out. So this could be called a basal respiration type test. Correlation was just as good with earthworm counts. This is not to say that soil CO2 caused the earthworms or the earthworms themselves caused all the CO2, but we're dealing with an infrastructure here of interrelated biological effects in the system. I want to show some very meaningful results when you compare different cropping systems. These are samples that were taken by NRCS in South Carolina and supplied to our laboratory based on the cropping history. And I've just indicated a few samples here. We had a truck farm that we sampled, continuous tillage farm, one of the lowest CO2 burst respirations I've seen in a long time. Occasionally we see numbers lower than this. If you compare that to a no-till topsoil, which was within the same county, same soil type, look at these large differences in respiration that we experienced. And even the subsoil in the no-till, under the no-till system, was significantly higher in respiration than the vegetable tillage system or bare tilled tobacco farm. This was different scenarios that were being examined of cover cropping. We had one of the highest respiration levels in a multi-species cover crop on that same soil type. Now, just as a comparison, I threw an example of virgin prairie soil from Nebraska in here, 78 parts per million on CO2 burst, exceeding all these. But what is exciting about all this is that we can obtain, with management practice changes, respiration levels comparable to virgin soils. Now, it's a little bit of an unfair comparison in that these are different soil types in different states. But if you do enough of these tests, you learn to spot these soils that have high respiration potential, and they are very productive soils. As an overall summary that I want to emphasize here, from the work I've done with CO2 respiration and the methods that we have developed, you have to look at CO2 respiration as a whole soil plant phenomena. There is reason to be concerned if we parameterize it too much into individual traits, and laboratory artifacts are something always to be concerned about, in that we cannot simulate the multi-diverse environment of diurnal moisture fluxes and temperature fluxes. So we have to agree on a standard for measurement, but we have to be willing to test it in field studies. One of the things that I continue to find astonishing is that biologically active soil furnishes a significant portion of photosynthetic CO2 demand. Based on early work I've seen that was partly reproduced in early studies in this country, there is reason to believe that crops could be CO2 limited under high growth conditions, meaning that there's another reason why you want a biologically fertile soil. Aside from the indicator status of CO2, it's actually a nutrient. The other fascinating thing about this, and again, there was a lot of emphasis on this early in the last century, and then it disappeared, but carbonic acid dissolved in the soil solution is a weathering agent, and it happens to be very convenient for plants because it solubilizes minerals at a slow and steady rate. In other words, the soil CO2 respiration system is itself extracting soils for plant mineral availability. The other thing I want to emphasize is I believe that the nitrogen, phosphorus, sulfur release from organic matter is quite predictable but it will depend on the structure of the molecules that are being decomposed. Therefore, we have to take into account, for example, the CN ratio when we are trying to predict nitrogen release. As I have tried to show here, a 24-hour soil test, such as the Solvita method, does provide meaningful correlations to field practices and is cost-effective at the commercial soil lab level. One thing I want to strongly emphasize here is that the influential factors, number one, are temperature. So we need to pay attention when we do these measurements as to what the temperature was during measurement. And when we do efflux studies in the field, we use recording thermometers so that we know the changes that have taken place during the system. I'm also very interested in correlating CO2 respiration with average soil temperatures in different growing regions. If we can then add on top of that the moisture variations in those regions, we can help determine whether basal respiration or CO2 burst is a primary driving factor in delivering nutrients. Most likely, I believe it is a combination of the two working together to provide this overall nutrient impact. I want to be finished with enough time to answer any questions about this and the cycling that I have addressed here and talked about. Again, I want to thank the sponsor, Ward Laboratories. Special thanks to our facilitator, moderator, Dan Davidson, and obviously to the Tri-Societies for helping make all these seminars possible. I think at this point, Dan, I'm going to turn it back over to you to coordinate or respond to any questions or discussions that would be available at this time.
Yes, Will. I've been forwarding you some questions through the chat box.
Yes.
Can you see those?
Yes.
You just want to kind of take them one by one?
Well, I have one question here about the difference between Solvita and Haney. I think if we could just look at this final diagram here, we developed the Solvita test to quantify microbial activity based on CO2 release and capture. As I indicated at the beginning of the presentation, there's a number of methods out there. You can pick your technology. All of them are valid measurement techniques. Some of them are obviously more costly than others, and some are really sample limited. When people talk about the Haney test, they're referring to a couple things on top of this. They're referring to the fact that we standardize this respiration as a CO2 burst method. Previously, our work had been emphasizing basal respiration. By standardizing it as a CO2 burst, we were able to bring it into the laboratory environment and quantify it, and this has brought it to the attention of ALP and NAPT soil proficiency programs, which are presently monitoring it. And I'm pleased to report that the proficiency is improving, and that as laboratories become more experienced with microbial testing, the variance between labs is declining. The other difference between the Solvita and the Haney test is Dr. Haney has also introduced a new soil extract. I say the extract is comparable to what I learned in Europe, such as the calcium lactate, lactic acid type extracts, where they were trying to mimic more soil environment type circumstances than would be the case with typically stronger, higher molarity extraction methods that are used in soil labs. So if you put all this together, you have one version of the soil health tool, the streamlining of CO2 respiration testing to make it suitable in the laboratory through the Solvita test, the standardizing of that, the requirement to weigh and measure the samples and quantify it, combined with this new approach to how nutrients are extracted.
Well, on the carbon to nitrogen ratios, when people think of wheat stubble and corn stalks and soybean stubble and other types of things, let's say alfalfa stubble, they have, huge ranges in carbon to nitrogen from, greater than 80 to 1 to down to less than 20 to 1. How is this affecting this soil respiration and the CO2 respiration?
Well, it's an indirect effect. The microbes are going to consume the organic matter with a dependency on the supply of nutrient, or in particular nitrogen, first of all, phosphorus and sulfur secondarily, to provide for building of cellular matter and protein and so on. So when we refer to immobilization, the respiration is going on, but the nitrogen is being sucked up and included in the microbial tissue until such point as the system reaches steady state, which could occur anywhere below a CN ratio of 15 down to 9 or 10, where those nutrients are no longer held by the organic matter and released. Now, I hear a lot these days about the rapidity of degradation of crop residues, and we are seeing farms that are reporting inhibition of decay of crop residues, and that concerns us a lot, because if we have a system being held back by low level of soil microbial activity, unable to decompose the organic matter rapidly enough, you can have a long, slow immobilization period there. And obviously, it's quite likely your CO2 release will be unusually low. Moisture obviously is implicated in this. If you have crop residue on the surface of the soil, only rainfall splashing soil and microbes up onto the tissue is going to bring about decay, in addition to what surface inhabiting worms will be able to affect. So it's all kind of tied together there.
If you were to take a soil sample, let's say 1st of October in Illinois, in central Illinois, after soybeans and after corn, considering the differences in carbon to nitrogen ratio, and what would one expect to see in terms of the CO2 respiration?
Well, you might not see a large difference in respiration, assuming the soils are biologically active and moisture is present, but you will see, if you did what we call a full soil health test, you will have evidence that there's a low level of nutrient mineralization going on under those circumstances. Now, from my experience in doing testing, we're seeing a lot of soils with moderate to high levels of soluble nitrogen at the end of the growing season. And obviously, if you plow stubble down into soil that has between 50 and 100 pounds an acre available nitrogen, there will be very little reason to believe you will have immobilization under those circumstances. That nitrogen will be quickly utilized. The beauty of the system is crop residue can immobilize nitrogen that should not be sitting there available late in the season, because you will lose that during the winter. It may end up in groundwater, or it may be denitrified. Obviously, the interest in cover crops at this time is partly to sequester these soluble forms of nutrients and keep them within the living plant tissue.
Here's a question that came in. Will, can you use a Solvita test, the Solvita test jar, and a paddle in the field, and turn the jar over on the paddle? How long should you leave it?
Well, that's what I would call a mini flux chamber, except when we do true flux chamber studies, we have a background flush of air that's introduced into it to try to simulate the real environment. We have found, and I want to point to the early work of Dr. Duran in Nebraska. He worked and developed the in situ test methods using a canister inverted over the soil, in which a detector would be used to capture the CO2. And these studies produced very meaningful data. Now, they were using gas chromatography and Draeger tubes in that, and towards the end of their project, they discovered our Solvita paddles and used them as well and had a very high degree of correlation with the Draeger and the GC approach to it. So theoretically, yes, you can do it. We are working right now on a field level type of test. It won't be the same kind of CO2 burst test to be used in the laboratory, but we're very concerned about the right sort of controls and methodology here so that people don't get just wildly different results. Obviously, you could put an inverted jar over some manure that might be in the field and you will have an aberrantly high CO2 rate. And inversely, if soil's been tilled and there's some subsoil exposed and you place a canister in that one location, you will experience a very low CO2 respiration rate. So what happens when you go into the field is you have to do a lot more careful sampling and be careful where you place your cylinders, and obviously protect them from sunlight, because if they heat up, it will create a significant CO2 flush just based on the Arrhenius reaction that I described earlier.
When does, another question here, during the course of the growing season in the Corn Belt, for example, when does the CO2 release, when does it peak out in the soil?
Well, that all depends on temperature and moisture. It's going, as I showed in some of the earlier slides, and I'm going to go back. Here we go. This is one of these again is Pennsylvania, and one is the Palouse, Washington, and you can see that if you ask the question here, where will you have peak CO2 respiration? It's not going to be here in this first diagram where you had the highest temperature, because you also have the lowest moisture. But when you have these rainfall events here where the moisture spikes upwards, you're going to have a big flush of CO2 respiration at those points. And Dr. Haney and I believe that if we could model these and put these into our forecasting, this would greatly sharpen our estimates of nitrogen mineralization from these kinds of systems. And clearly, here you have even greater peaks. Here we have late August and almost no soil respiration taking place under these dry conditions. This is dry soil. When the moisture returns, you will have a significant off-charge, and as I suggested earlier, there's a considerable body of work being presented now on CO2 burst phenomena because it relates to climate effects from soil warming. The CO2 burst event makes up for the period of time that the soil was suboptimal in moisture. In other words, we believe that the enzymes have continued to accumulate and stay active in dry soil, and when the moisture returns, the system compensates for the dry period, and you get a nutrient charge. So theoretically, such as in the example here of the Palouse soil, this return of rainfall towards the end of the summer will bring a last charge of nutrients into the system.
I have a question. I have a question. Well, the NRCS promotes measuring soil quality because you can't have health without good quality. And so, measuring, and this is in one of their suggestions, you do the basal Solvita test. So you do the basal Solvita test, but I don't know how now after your presentation, and I'm wondering how meaningful that is, because you're out in the field, you're going to look at the soil quality, you're going to do 10 different tests at one time, and in the middle of the day, it could be dry, could be cold, could be hot. How do you interpret that number from the basal test? I don't know, maybe it's misleading. That number could be misleading because just based on sampling error.
Well, you could say the same about lab testing, because laboratory testing is notorious for being an artifact of a system. And if you work with field ecologists, as I do often, they try to avoid the lab, and the laboratory people are avoiding the field workers because they both have objections to the parameters and the variance associated with them. Basal respiration is a wonderfully realistic way to look at the system. It's leaving it in place, or we advocate minimal sampling. In other words, take a sample of soil, don't mix it too much, you remove sticks and stones, yes, leave it moist. You can do repeat measurements under those basal circumstances and get very reliable indication of the soil under the temperature conditions in which you conduct the test. As I said earlier, you may have to do more sampling. You have to take notes about where you place your samples or take them. But these results, if connected to the crop CO2 demand, which I started this presentation with, I believe basal respiration is the future, because that's going to define whether crops are getting sufficient CO2 out of the soil system. The problem is we have to take into account diurnal temperature, which is the flux during the day, and be able to measure it meaningfully and project that daily factor. So yes, I believe it can be accurate. Soil testing in the laboratory is considered to be more precise than field testing, and yet from another perspective, because it's performed under less realistic conditions, drying, hammer milling, sieving, and so on, you could argue that you have lost some of the features of a true soil quality system.
One additional question on that, Will, because I think about doing soil quality tests myself. So on this basal test, should you do them like at 9 in the morning, noon, 3, or 6 in the evening, so you reduce the heat load? Does that make any difference?
Well, again, keep in mind what I said about the temperature of the system. If you bring, I live in a region here, for example, where during the growing season, probably the average temperature of the soil for the entire growing season is probably 17, 18 Celsius. So you could argue that bringing the soils in and running them at that temperature would represent seasonal basal respiration most realistically. If we incubated them at 30 or 35 Celsius, we would get more CO2 out, but how realistic would that be when projected to the actual circumstances? So in the chart and diagram that we have on the screen presently, you can see you have to choose a point here between the rise in temperature in the spring and the drop in temperature in the fall, and find out what is the average circumstance. Maybe measuring the system under those conditions would be the most realistic. Measuring at 20 to 25, which we do now, is in this upper quartile of soil activity. Possibly one could argue it's unrealistically high and would overrepresent the system, but we're all using this as indicators, and we're correlating it with crop response. So that has to be kept in mind.
Okay, all right. Well, we're at going about 8:05, 8:04 p.m. Central Time. We want to bring this to a close, this 1-hour webinar. Again, I want to thank Dr. Brinton for this presentation tonight, and talking about some of the factors about CO2 respiration and how important that is to the crop and to mineralization. Again, I want to put a word in for Ward Laboratories in Kearney, Nebraska for sponsoring this event, and so I want to thank them. And again, as a reminder, that Odette Menard webinar that was not held last week will be held on Monday night, July 7th at 7 p.m. Central Time. You should have got an updated announcement by email today. Again, thank you very much for your time tonight and for sitting through this webinar. This presentation will be online in a few weeks. I have recorded it, and in a week or two, the American Society of Agronomy will post it online. You can come and listen to it again, or any other people that couldn't make it tonight could listen to it as well. So again, I want to thank you for your time tonight, and